I would also like to welcome you in the name of the Food Packaging Forum. And I will just give a short presentation and try to set the scene and try to cover not all the aspects we are going to talk about today, but maybe some of the aspects, and I try to be simple. So you will hear more details later in the further talks. So the title is Food Packaging and Chemical Safety Today. That is a very short part, and then I will tell you something about the scientific challenges as we have identified them about the food safety and food packaging. So let me start with some numbers. Here you see the food packaging market, the global market, which is worth at the moment $270,000 billion. It's an estimate, and it's going to increase in future. So it is indeed a very important market and an important issue we are talking about. Almost everybody uses food packaging, and it's a very valid thing or valuable thing because it's protecting our food and it's preventing it from food spoilage. You can use it for longer, you can transport it more easily, it's practical, all of us know the advantages. But the topic we are talking about today is also the chemical safety, and there are some challenges we may have to face in future and are already facing at the moment. So what do people think about the chemical safety of food packaging? I was wondering when I started to prepare this talk, and I found two surveys. One was done at an open house at JRC, and the other one was an online survey, which is just from last year. And people do worry about the chemical safety of food packaging. They really think, okay, there are chemicals which may harm my health. There may be something inside which I don't want to be there. And similar results came out, maybe not as high, last year, that more than half of the people who were asked do worry a lot or to some extent about the safety of food packaging related to chemical contaminations. Now, how do we assess the safety of food packaging? And we go back to somebody who was born just very close to here in Switzerland more than 500 years ago, to Paracelsus, and we usually check whether we find safe doses of chemicals in our food, in that case, and see if they do not harm our health. And usually this is done by preparing these dose effect curves, as you see here, just an example. I'm sorry. And that should be done for most of the chemicals, and then you could define a dose which tells you this should be safe or safe enough. To show you a mathematical term, you can calculate the risk of a certain chemical by knowing the exposure and knowing the hazard of this chemical. And I will come back to this equation quite some more time later on. Because we think it's although it looks simple, it's not a very simple equation really to solve. And the main challenge we have identified are just the number of substances we are dealing with. So I tried to categorize them, and we had discussions about the definitions of these substances just this week again. So I have the first category that are the intentionally added substances. So that are all chemicals or chemical mixtures which you add to produce a food packaging or also a food contact material, which includes many more materials or things like conveyor belts and big tanks and everything which is in contact with your food during the whole chain. There it's already hard to find numbers. You find them, but you don't know how much of it is used. Just to show you some numbers we have extracted from lists. So there are between 900 and 1,000 entries in the union list. So the positive list, which is used to produce plastic. Then there are approximately 6,500 entries in the FACET database, which is the European database used to model and calculate migration of chemicals into food from FCMS, and also including other topics, but these are just the real food contact material substances. Then there is an American list containing direct and indirect food additives with more than 7,000 chemicals. And then there is a Swiss list which contains even more than 500 substances which can be legally used for the production of packaging inks. So you see it's a huge number, and not all of these chemicals are used, but there are still many on these lists. And there are even more, because these chemicals are the starting substances. So these are the ones which you use in the beginning when you produce your final material. Maybe they are not even there anymore, but maybe they are converted to something completely different. Or maybe they are not pure in the beginning, so you have impurities already which you don't know. And all these other chemicals can be grouped into the non-intentionally added substances, or NIAs as we call them. So they contain the side products which occur during the production of most of the materials. They contain the impurities. They also contain breakdown products, and not only the non-intentional breakdown products, but also those which are break down on purpose, like UV stabilizers. They are designed to break down during use. And they also contain contaminations from, for example, recycling processes. And I found data published by Connie Grob, who says that almost 100%, so far more than 90% of the migrate from can coatings is indeed a NIAS and not the starting substance. So it's not the BPA which is migrating, but all the derivatives which are derived from the polymerization reaction. And then another set of data, that's not a migrate, that's an extract of polypropylene, says that 60 to 90% of the chemicals which can be extracted from this plastic are NIAS. So this shows us that there is a big part of the chemicals we really don't know how to handle them. So let's get back to this simple equation. So even if you think it's a simple equation, we need to have the data to solve the equation for all the chemicals we are using. And there are only less than 2,000 starting substances authorized in Europe and have a valid toxicological evaluation. And the NIAs, I've already said that they can have different sources, but they can also have different qualities. So they can be known. Maybe you evaluate your migrate and you see a chemical which you even expected. So you know the chemical structure and you even know that it has been evaluated, so you can more or less easily handle it. But maybe they are not known. Maybe they are known but not tested. So you have a structure, then you're already lucky, but you don't have any idea on the toxicity of this substance and on the exposure of this substance. And then very often you detect peaks, but you don't know what the peaks are, so you cannot easily identify them. And this is a more theoretical case, but I think it's still there. You may not even see them with your analytical method you have chosen. Then I would like to question the first term of this equation, so the exposure part. Exposure from FCMS is not direct exposure. When you compare it, for example, to food additives. If you have a food additive, you know what you add to the food, and you can calculate how much the population will eat of this chemical. But migration is the step in between. So you have to see what happens, what interactions happen between the material, your food, maybe the outside of the food, that you have to somehow predict. So that's also challenging. Then the second point I would like to make regarding exposure is the cumulative exposure. If you have good data on the exposure of one certain chemical, you have to consider that this chemical might have other sources. So it can come from other products you're consuming or you are exposed to, and you can have oral intake, or you can have dermal uptake, or you can breathe it in. So there are different exposure routes also. That has to be at least kept in mind when you do an exposure assessment. Now I want to focus, and that's, I think, something we are really interested in. Now I would like to focus on the second part of the term, the hazard. And the hazard characterization is even more difficult, I would say. And I just show you three concepts which we think have to be considered, but as Martin said in the beginning, we cannot present you solutions at the moment, but we think it's important to keep them in mind. So the first part is the mixture toxicity, and I just show you a very simple example. All of you probably know that we are exposed to highly complex mixtures. And just to show you what can happen if you're exposed to mixtures, I've chosen this one. It's a mixture of triclosan, which has been used in the area of FCMS. It's not authorized at the moment. It's a biocide. And then the second is it's a pharmaceutical ethinyl estradiol, the active ingredient in the pill. So if you feed rats with ethinyl estradiol compared to any controls, now if you feed them with triclosan, compared to the control, you don't see any effect in uterine weight, which is one bioassay you can use for estrogenic effects. And you don't see any significant effect when you look at the expression of estrogen receptors in the animals. If you use ethinyl estradiol, you see an effect. That's what you expect because you know it's an estrogen. But if you use both at the same concentrations, you increase or decrease the effect. So you enhance at least the effect which you have observed only with the hormone. And that's a very simple example. And if you imagine to do that with 100 chemicals, it's getting really difficult in the end. But we have to think about it in future. Then the second part I would like to focus on are the non-monotonic dose responses. That's again related to hormones. And it's really challenging the dose makes the poison sentence. Because there you can assume if you have a not linear, but monotonic response, then you can somehow extrapolate a safe level. But what has been observed for many chemicals, and in many cases, I've chosen just from different areas some examples, this is not always the case. So on the x-axis, you always see that here the or the tested ingredient in different bioassays has been changed or has been increased. So there is always an increase from the left to the right. And it's always tested for the same endpoint on the y-axis. So you see that these dose response curves are not linear. So what happens if you would measure somewhere here and then you don't measure anything here? You cannot extrapolate any effect. So that's something which is also difficult and has to be assessed, especially for substances which are supposed to be endocrine active. Then the third point I would like to make are the so-called sensitive windows of development, and they relate to the time of exposure. We are not, so usually in a risk, or very often in a risk assessment, it's assumed that you just take the same amount all over your life. But the same amount of a chemical may harm you when you are a small baby, but it may not harm you when you are an adult. So there are these sensitive windows of development, and they have been defined as mainly the first 1,000 days of life, so during gestation, infancy, and early childhood. And if you're exposed to a certain chemical, that can have an effect on your later life. So you don't become ill immediately, but you may develop a disease later on. And there are quite many examples already out there that this can or does happen. And then just this year, a second sensitive window of development was identified, and that's again related to pregnancy, but it's not the child which is sensitive at that stage, but it's the mother who is. So the mother, that was an animal test, so it's not proven in humans, and it's also difficult to prove this in humans. But there are good animal models existing. So the mother of rats or mice, that you can check in the reference, of mice, I just hear it from the audience, developed diabetes when they were exposed to BPA during pregnancy, after pregnancy. So compared to the controls who were healthy afterwards, the mothers developed diabetes. So that's quite striking that even after pregnancy and you think you're grown up and developed, that you can still be so vulnerable to a certain chemical. So that were the missing arrows here. So again, let's come back to the simple equation. So first I just said we don't have the data, but we have other things which we have to integrate into this equation. So it's the migration and the whole exposure issue. Then the mixtures, which are of course related to exposure because you need to know the amount of chemicals and the differences of, or the mixtures of chemicals you're exposed to, and they can even result in a completely different hazard. Then the sensitive windows of development have to be considered when you do a hazard identification. The non-monotonic dose response curves. And what I didn't mention here, but I think it's also very important that you simply choose good toxicological endpoints for your hazard assessment. And then another thing which I didn't mention in detail, but it goes into both, you need to have uncertainty factors because you cannot do the test with a human. So you have to somehow estimate how we can transfer your data to the human population. So who shall solve this problem? Our aim, as we defined it, are safe food contact materials. And we just made this little star here and would like to add that that should be based on most current scientific understanding. That's something we are really working for. So I think all of us have to work on this, and all of you. So it's a mixture of all different, again, a mixture of all different people. So there are the scientists working on the materials, on the analytics, toxicologists, epidemiologists have to be included in this. Then the authorities, the regulatory authorities and the enforcement labs should work together with everybody else. And then last but not least, the FCM manufacturers and also the food industry, who at the end is responsible for the safety of the packaging or the food contact material which they are selling. This was the wrong direction. So we need good cooperation, and we hope that with events as we have today, we can enable this communication and start a good dialogue. And during the year, we hope that we can provide you with good information also on all these topics which I just mentioned. So let me conclude. We think the data availability is not sufficient at the moment to guarantee safety. That's sad, but I think it has to be said so clearly. Then we have to integrate new scientific understanding to the classical risk assessment. I don't want to say that stop measuring those response curves. Of course, it's highly important, but when an experiment is designed, other things have to be included. Then there are already testing methods which do include some of these new scientific understandings, although they may not be regulatory requirements to use them, but it would be maybe interesting to think about using them already. And the final point, there are clear strategies needed and open-minded discussions are needed to integrate all these issues and to further increase the safety of FCMs. So thank you. With that, I would like to finish.